Okay, let's start our second part. Now we have discussed epigenetics part, how histomodification associated with binding to gene and how to affect it to gene expression and also transcription factor binding, how we use NGS technology to identify modification, to evaluate the modification and identify transcription factor binding site. But we still have an important part about the DNA methylation. In next one, we will talk about what's DNA methylation and how can we test the DNA methylation and what's the effect of DNA methylation with the transcription factor with the transcription. And the DNA methylation is we can say there is a we can see here, for example, this is a cytosine is methylated. And in this one, people, in the gene regulation, people are most interested in the CpG methylation. CpG methylation, we know it's can spontaneous deamylated to form a T, C2T, and also just because DNA repair systems, they poorly recognize this methylated CpG. So for a long time, and that CpG can eventually become TG mutation, then propagate it. It's the most common case in some cancers. And but for DNA methylation, people usually know DNA methylation can repress downstream gene transcription. People already know DNA methylation is essential during the embryonic development, and people and in few years ago, and the people still only found very few transcription factors combined to methylated DNA. For example, like MECP2, MBD102, or few single proteins. They can binding not a sequence-specific methylated DNA. And as I mentioned, the DNA methylation in promoter region, they usually repress downstream gene transcription. And the aberrant DNA methylation pattern are associated with a number of human cancers, resulting global reprogramming in gene expression. Here is one picture shows and how DNA methylation can affect the transcription factor binding. One scenario is we know here is a promoter region. If there is a unmethylated CpG here, unmethylated, you can see, and the border here, and then transcription factor, if they recognize some sequence here, motif here, they can bind here, then active downstream gene expression. However, if those CpG sites are methylated, they can either, because they methylated C, they cannot be recognized by some transcription factor. So transcription factor won't come here. They won't bind here. That's one scenario. Then gene will be silenced. Another one is just because the CpG site is methylated, they can recruit MBD protein to binding here. Then they occupy the site to block other transcription factor from coming. Then gene will be silent. That's most of the two scenarios. And before doing that, we want to know what's the CpG island in the promoter region. As I mentioned, in the human genome, you usually have 42% of GC contents. So if we say, okay, you can get the simple status count. If 42% GC, which each one is evenly like 21%, then for the CpG dinucleotide, then we have a 4% frequency of that one. But unfortunately, in the human whole genome, is very significantly depleted. It's only 1% of CpG. However, in some area, especially in promoter region, we can find the high frequency of CpG site, which we call the CpG island. How can we determine one region is a CpG island or not? There is a three criteria we used. The first one is the region length. We want the region to be big enough, not only one or two CpG sites. We want like about at least 200 base pair. And also GC contents should be larger than half of that. And then we have observed to expected CBG ratio is greater than 60%. If you don't want, you don't need to know this formula here, but just understand that doesn't mean, for example, if we have one area, they all CBG, and can we say is a CBG island? Maybe not. And because the observed to expected CBG ratio is not high enough, it's defined as a number of CBG is divided by expectation. Expectation can be calculated by two ways. One is a frequency of C or G, or the other one is C plus G, average of C plus G, then divided by the length. Then if the ratio observed to expected ratio is larger than 60%, and also GC count is larger than 50%, the region is big enough, we call it CBG island. CBG island, why the concept is important? It's because people know CBG island are usually associated with genes, especially in some housekeeping genes. And they are typically common close to transcription start site and in the promoter regions. And normally, a C immediately followed by G, there is rare in or the bad DNA because the C in such an arrangement tend to be methylated. And so which means CBG island here is a picture shows this is a gene body, this is a promoter region, and there is a high density of CBG here. And we assume there are CBG island here, but most recently, we have a more sexy concept we call the CBG shore. It's something like around the CBG island, like 2,000 base pair away from CBG island. And some people also call the more extended area as a CBG shelves. It's a 2,000 base pair away from CBG shore. But it's just a concept that you just keep in mind. It's not necessary to pay much attention there. But however, CBG shore people prove is most important, especially the methylation and the CBG shores. CBG shores are more tissue specific. For example, this is one paper published in Nature Genetics in 2009 or some year around 2009. And here shows an example like a cross tooth. This is a CBG core, and this is a CBG shore, and they compare DNA methylation from 3 tissues: brain, liver, and the spleen. And you can see in the CBG island, almost the different DNA methylation is quite similar, but you can see a diverse, distinguished pattern around the CBG shore. So they claim CBG shore, the methylation at the CBG island shore is a tissue specific. Now we know, and this is a whole pattern about the gene regulation. We can use ChIP-seq to identify transcription factor binding site. But now, and is any similar, the ChIP-seq, the main idea again is to pick up the sequence of interest or transcription factor binding. Then we see what they are. Is there any possibility we use some technology to pick up methylated DNA sequence or unmethylated sequence DNA? Let's review ChIP-chip or ChIP-seq first. And first, we get the genome DNA. Then they do cross-link and share, and then get a number of DNA. And then how we can extract the sequence of our interest. I reminded you last time, what is the most important factor in ChIP-seq? Antibody, right, antibody. So people may think, okay, let's ChIP-seq can get transcription factor binding. But if we can find an antibody associated with methylation, we also can get those sequence methylated, right? Or some antibody with unmethylated sequence, we can get those ones. So here is another one example. which called the MEDIP microarray, very popular in the early year. And the full name of this technology is methylated DNA IP. So this pattern is quite similar to chip. And first we have a control here, and then we have a sonicated genomic DNA sequence here. Then we use the antibody, IP DNA with a monoclonal antibody. Space will recognize the 5MC. Then pick up those methylated one. Then we label them with different color. Then hybridize it or sequenced to try to see how much of that. And the bioinformatics analysis on that one is okay, but we have some kind of a challenge there. First one is we need to know the bias, or we need to normalize bias in ratios, because the bias comes from like a proper sequence or GC density or intensity. Here intensity we got doesn't tell you exact methylated level. They just tell you the relative methylation level compared to control. And also, because this is the probe, and they have a probe. So the methylation level is the region of the probe. It's not a single nuclear base pair. Of course, the similar principle can use for others if we can find a different antibody for that one. For MEDIP, so as we mentioned, is that they use a monoclonal antibody specific to 5MC, but still they have other ones called MBD, affinity purification. So the ipDNA containing methylated CpG using an MBD column, or they have something, it's opposite to that one. The ipDNA containing unmethylated CpG use a CXXC domain columns. And then the idea is pretty similar, and they just pick up those ones, or they may focus on some restricted region by use particular enzyme they digestion. For example, they use cytosine sensitive enzymes followed by whatever they called, and it's another technology. They try to get a restricted region, not the whole genome, but the challenge of those technologies, the very small number of cytosine sensitive site can be achieved. I don't remember the number, but I know it's a very small number. So that's the reason why people switch to other DNA methylation experiment. And then nothing I have to mention is that this method is very in their ability to detect the differential methylated region between pairs of samples. Now, right now, there is most popular technology we call the bisulfite sequence. Bisulfite sequence is we use bisulfite treatment and sodium bisulfite sequence to convert cytosine residue from C to T, but leave those methylated one to C. You can see here is unmethylated C. Then by bisulfite, they convert to U here. But after PCR, they become T. And the corresponding anti-strand sequence also changed to A, from G to A. So theoretically, this technology can, then we do sequence. So theoretically, this technology can map the entire DNA methylome in one base pair region. We can know for each base pair, and CpG side is methylated or unmethylated. It's good, the bisulfite sequencer, but the large scale, especially for genome-wide bisulfite sequencer, they have been successfully conducted, but they have some challenge. First, they require full genome sequence or control in the bisulfite chip samples, which is time-consuming and also some resource-intensive. And the cost is higher. Of course, why the sequence technology, they decrease the price so we can get a maybe reasonable cost price right now. But also, I have to mention, bioinformatics analysis is a little bit challenging until people develop new tools right now. And another thing we have to keep in mind is the convolution by software convolution sometimes is not quite complete. And also they may degrade the DNA during by software treatment, which may bring false positive or false negative. And also you may mention, okay, I don't need the whole genome. I just interested in some restricted region. Then there is another technology. They use they use MSPL dissection. They use this methylation sensitive restriction enzyme to do dissection, and then do very limited regions of their interest, and then do by software sequencing and try to get what the cost is much lower, but of course it's not a whole genome. It's just for some area of your space, of your particular interest. I mentioned there is a challenge in bioinformatics analysis on by software sequence. What's that? Now let's first take a look at sequence alignment. Here I showed, for example, I just use a simple example to explain that. This is a reference genome. And if you test the sequence you need to test is like this one, then you can get a fragment by sequence and then match to the genome, try to find their genome location, right? But however, for by software sequence, first, this is a sequence, and this C, the red one, is a methylated, and the blue one is unmethylated, and this is an anti-strain and complementary strain, and you can see, and this is C, in the complementary strain, C is unmethylated, this 2C. However, when you do by software sequence, the first one can be this C to T, and this 2C to T, but for methylated C, they keep there. For the complementary strain, and this C to T, and this C to T, and after PCR, we may get those from each one, we may get some sequence like this one. I already convert them from 5' to 3'. And then when you do mapping, you find something doesn't match. It's here, TC, TAAG, TCT. It's first T is not C. And then even from another string, you may find that here become A. And more example here, this become TT and this A. Then how can you do that? How can you map them to the genome? Can you say, okay, I allow, if I say the first case is exact mapping, then can I allow some mismatch? Can I do that? Of course, if you want, you can try, but I 100% believe there is a more other sequence. They are not exact match to the whole genome, maybe coming here. Now, what do people do? There is a software or there is a tool they developed called by Bismarck, by Barbara Ham, by Informatics. And this Bismarck is a tool to map by software convoluted sequence risk. Then they can calculate the cytosine methylation status. And it's written in Perl. And there they shows how can you do that. This is a nice website. But what's the main idea of Bismarck? The main idea is a Bismarck. They need to change the genome, reference genome. They have a four reference genomes because they did a pre-convoluted form of a genome. They C to T or G to A. And then, like this one, this is a reference genome. Okay, this is the original, where's the original one? I think this is maybe it's the same as the original reference. Then they have a 4 type of convert. Then for the older sequence after by software, they also transfer all C to G and G to A. But still, which one we should compare? We have no idea. But because we don't know the strain information. We don't know from which one, right? So for C to D, they need to compare C to D, C to D. And for the G to A, they also need to compare for read strains. They need to compare to the all four reads and the four possible alignment that they have to do, like CD converted reads to CD converted, and the GC converted, and also to CD converted, and the CD converted also to GA converted, and the GA converted to GA converted. Sounds weird, right? But just keep in mind what they do is first they need to make 3 additional reference genome and do the conversion C to D or G to O C to D and O G to A. And then they need to do for the sequencer and they need to test that they also have to do similar conversion. Then they compare to all the one. Then they can find the unique mapping one, the best alignment. And then from that one, they also can find the strain information. That's the most important mark for the Bismarck. So here is a workflow of the Bismarck. First, you need to have to do genome preparation. Of course, you don't need to do that manually. They can show you how to do that. And also, you just need to do once for each genome, like a human or mouse. And then next time, you can start from second step, Bismarck alignment, and then they put the Bismarck alignment and the methylation call together. Of course, we strongly recommend you to do fast QC before you do anything. You have to remove those sequences with a low quality. Otherwise, you may get many false positives. And after the alignment and the methylation call, you can get 2 output files. One is a BAM file, including alignment and also methylation call strings. Then we put those those C information there to show you what is it methylated or unmethylated, and is a CHG methylated or is a CHG methylated or is a CPG or is a CHH. And you can find the manual to read the manual to try to find all the information. And also they have another text file as an output, including the alignment and the methylation summary. I will show you next slides. And with this one, then you can use a Bismarck methylation extractor, then get all the CPG side or all CHG side, all CHH side for their methylation level. The summary report of Bismarck shows how many numbers of C analyzed and how many methylated C in non-CPG or in CPG, and the total C to T conversion in non-CPG or in CPG, and also C methylated but not in CPG, C methylated in CPG. So you can get such information like the rate of methylated C or convert rate for anything you want. Then from Bismarck, you can get all of them. You can, which is the software is very powerful. You will like it, and it's very straightforward and easy to use. I guarantee that. And okay, we have time now. I will talk about the one story as the last part to how to use all the technology or most of technology to have a comprehensive study, or maybe we can call crosstalk between DNA methylation, histone modification, and the transcription factor, and the transcription factor regulation. As I mentioned, people found DNA methylation and the anti-correlation or negative correlation between DNA methylation and the gene expression. Here, for example, here is the difference of DNA methylation in 2 tissues, liver and brain, published as I mentioned before. And here is a gene expression difference. And most of them, you can see, if the difference is negative, difference of methylation negative, and the difference of gene expression is positive. So which means it's a negative correlated. However, don't forget those portions. Are they noise or no? Then we can look at another study by our group. We also compare, but here is in mouse, brain, and the retina. And we also look at the DNA methylation and also gene expression. For this one, it's quite similar. You can see high DNA methylation. This is a two tissue, brain, mouse, retina. So for high DNA methylation, low gene expression. For low DNA methylation, which is a high gene expression somewhere. And then, but we can find a portion of that high DNA methylation and high gene expression. Maybe this scatter product can show more detail, more clearly. And you can see most portion, about the 2/4 of DNA methylation and the gene expression, they show negative correlated. But still, we can find 1/3 of such pair, they are positive correlated. For example, we have one group here, we show DNA methylation level cross this gene, we call it whatever. And you don't need to care about what's the gene name, but we know in the promoter region. And we have a retina in red, the DNA methylation, and the brain in green, in blue. And we can see the difference. And the blue has the brain has a higher DNA methylation, but we found that the gene expression is higher too. So which means this part is a positive correlated. So what's that mean? That could be why we can see some such a positive correlation. Let's think about the first one. Let's think about the conventional pattern. Like we know here is a DNA binding site. If it's a methylated C, then which will block transcription factor. But let's say, okay, the other one we just showed is a positive regulation. So it give us a hint. If it's a unmethylated, maybe no transcription can come here. Only if the CpG site is methylated, some transcription factor may recognize this site, then binding here, then active downstream gene, right? That could be one scenario. So we collaborated with one group in Hopkins, and we try to see how many transcription factors can bind the methylated DNA sequence. The most important one here, or one of our interest in the KF4, and I want to tell you details about that, but you can just show you result. You can see KF4 can bind the methylated CCC, GCC sequence here. But for unmethylated one, they bind a totally different sequence. And why we care about the KF4? KF4 is a pioneer sequence. It's a member of KFM family and the transcription factor family. They can regulate the proliferation, differentiation, apoptosis, or somatic cell programming. And from other study, from another study using KF4 ChIP-seq, and also we integrate with a mesothelin data in the same cell line. We also use bioinformatics to by informatics analysis. We also identify some KF4 binding site. They are highly methylated. The motif in their ChIP-seq binding site is pretty similar to what we identified in vitro. So it gives us a strong confidence to follow up to see what's the function of KF4. And then we collaborate with another group to try to see KF4 in glioblastoma, because we know KF4 is a cancer driver gene. They're showing upregulated in glioblastoma, and the loss of genes will be associated with this one. And in previous publication, as I mentioned, KF4 can bind in two different sequences with a different methylation status. One is a methylated one, the other one is an unmethylated sequence. The binding domain are totally different and independent. So in other words, we can mutate one binding domain, but still keep another one. So our collaborator was very excited about this finding. So they engineered two doxy-induced human GPM stable cell lines, which each one they can carry KF4 wild type, which one, or mutated one. Mutated one means that we block the methylated binding site. the Y type one means we have both one. But then they find that the cell type is very different. The phenotype is very different. And what we do, but first we want to know what's the target of care4 method of binding. So if we want to do exact target, especially in terms of gene expression, definitely we will do iron-seq. We try to see which one is upregulated or which one is downregulated, and what's the association between those ones with the care4 chip-seq. We want to know, for example, here is one example, ROC, and you can see care4, there is a 2 care4 chip-seq binding here, only for Y type. If we block mutated, they are methylated binding, no peak here. So which means those two, they are associated with care4 methylated binding. And also we tested this one, the ROC gene expression, and we can see if we have if in the Y type we keep the care4 methylated binding, we can see the ROC, this gene can be upregulated after 48 hours. But if we block this methylated binding, the gene expression doesn't change. So which means, which indicates this ROC is most likely to be target of care4 methylated binding. Cross whole genome, and we also try to find, we use bisulfite sequence to identify those methylated binding site, and then we do motif analysis. We get a motif. You can see this motif is quite similar to what we identified previous. But still, this is some target gene we can identify, we can define, because right now the target we define is a care4 has a binding motif in the promoter region. But we need to know what's the enhancer, what the enhancer target. So we use a chip-seq to test H3K27 agitation and to see which signal coming with care4 Y-type specific binding peak together. Then we can identify those regions, and then we say, okay, those are enhancer region. And then by informatics prediction, we can try to see the target of those enhancer region. Then we can get a number of, we can predict 100 over 100 care4 methylated active target. You don't need to care about the function of those one, but just to show you those targets are significantly enriched in regulation of cell communication, morphogenesis, adhesion, something strongly associated with the glioblastoma. But still, we are interested how this transcription factor, this care4 methylated binding can affect acylation signal change. And if we can see, if we mutated methylated binding, almost from 0 hour to 48 hour, acylation signal doesn't change at all. But only if we use this Y type, and we can see after 48 hours, we may have much more acylation signal. So this may give us a pattern. What kind of a pattern? The pattern is, we assume, okay, in the first beginning, this gene, for example, ROC is not activated because there are so many methylation, is highly methylated in its promoter region. And also the histone may be very dense, condensed. So there is no way to be activated until we have some care4, Y-type care4 coming, which has a methylated binding motif. So they can recognize somewhere, sequence match the motif site. Then they can recruit acylation signal, then unwrap those histone, and then active downstream gene. This is a very exciting thing to biologists, even though even at the first beginning, I didn't realize that, but very exciting. But still, I can see from your face, you already lost, I know, but I just want to show you. And what we do, we use iron-seq to can find some gene expression change. And then we can do chip-seq for transcription factor, for example, care4 here. And then we try to make a connection between these two data And then we know which one regulates which one. What's the target of that one? And also maybe we can recruit by sulfur sequence, try to identify those methylated binding, or for the binding site we are interested. Or then we can do more ChIP-seq for histone modification. Then we can get a connection each other. And from that one, you can say, okay, I use the enhancer prediction, I use the motif sequence analysis, or all the bioinformatics analysis, we integrate all data together. It's like what? It's like a puzzle game. Now, after that, you may get some puzzle, either beauty or beast, whatever you get, but then you are happy, right? Like me, I was excited when they tell me the importance of those biological questions. Yeah. So this gives us, this is just one example. Doesn't show you how important the biological questions, but show you as a bioinformatician how to understand the biological question and then try to find some technology to either prove their hypothesis or reject their hypothesis. Okay. Now, I try to finish my part first, then give time to Sean. The last part is a hands-on practice. Sean, and first, I'd like to thank two people here, and Dr. Shen Liu and Dr. Sean Courtney. And they did much work about the hands-on practice. And the hands-on practice is focused on MAX2 today. And we try to use MAX2 to identify the narrow peak or broad peak and to see, based on the ChIP-seq data and see. And here is what I explained the last time about the format of MAX2 output. I leave Sean to finish this part. I don't go through that one, but I just wanted to show you something I underlined the last time. One thing is, do we need control or not? We selected some database and some data from ENCODE. I didn't mention here. Then, for example, first one, I choose one transcription factor binding ChIP-seq is a CTCF ChIP-seq. And Shen used the two max call, negative call, try to identify one is without the control, the other one is with control. Then we will see what's the difference. This picture shows the raw data here, and this is a CTCF ChIP-seq, the top one. The bottom one is control, and without control, and we can identify two binding sites, this one and that one. If you don't look at the control, you can see those sides. Yes, it looks very good and higher than others, even though this one maybe just around the cutoff, and these two are significant enough. But when you look at that one, even by eyeball, you may doubt, what's that? And what's that? And from that one, you can see, okay, if I enlarge the first area without control, the risk looks good, and there is some risk pile up here. But when we look at the control, there are some enrichment here too. Even though we have no idea what are they, maybe some bias or somewhere, but definitely we cannot call this as a P. So this is a false positive, which can be identified if we use control. For another one, even we see here there is a bias on this region, but we can see the ChIP signal has a much higher signal. So maybe it's a true peak, unless we know something happened here, but we prefer to think that this is a true peak. Another thing is I mentioned is a narrow peak or broad peak. Here we use another example, H3K9 trimethylation. As I introduced it before, H3K9 trimethylation is a histone modification, which usually has a very broad binding region. So from that one, if we use the narrow peak calling, we only see here, this is a significant compared to control. But however, if we use a broad peak calling, we can see a true binding region, which is much broader than narrow peak. And when you use a broad peak calling, it will always give you several files, including one is a broad peak output, the other one is they call a gapped peak. Gapped peak file, they include some information like chromosome, star, and which is similar as the other output, and the name, and this is something proportional of 10 times of log, I think log 2 Q value. Yeah, it's a log 2 Q value here. It's a 10 times that one because they want to show in usage as a genome browser. And here shows the star and is the same as this one. Some information important one is maybe you are interested in that they can show you how many blocks in here. What means blocks? It's more like it's a broad region they called, but still maybe you can find something. Do you remember last time I mentioned for Max2, they allow some gap. So if they have a gap there, but they will mention, okay, we have 2. So for this one, they also show you what's the start, the length of each one, the first one, the second one, and what's the start position. So I pick up the top one to show you how they look like. It looks like this is the whole region for the first one, but you can see they have 2 blocks. Between them, there is a gap here. So you just need to understand the output, what they mentioned. So in future, maybe you are interested in some region, they have multiple blocks or something, or you have doubted about those ones. You can go back to check that based on this gapped peak. Yeah. I'm done. Yeah, I know you are happy to hear that. So, and take a 10 minutes break. Then Sean will show you one by one to give you more details how to do Max2. And again, and told me, and this lecture, we don't expect you know everything in the lecture, but we want you remember and understand the main concept there, like the difference of technology, or at least when I should use which one. And also as bioinformatician, for different technology like chip-seq, when should I call narrow-peak? When should I call broad-peak? And if my collaborator, they don't have a control, what should I tell them? Something like that. And I wish I could give you example about the final test of this part, but unfortunately I couldn't. But maybe, but I believe everything we will test you already in the slides. And if you understand them, so you should be okay. No problem at all. So good luck. I really appreciate your patience. Yeah. Okay, thanks.